Record. All right. Hey everyone, welcome to the first exam review. So this is fun stuff. We have a lot of slides to go through. We have like 60 slides. We're probably not going to go in depth for everything, but we're going to do our best to cover anything you guys are uncomfortable with or want to go over. We know it's a lot of material, so we put lots of memes and other things, other fun surprises throughout the slide to keep it light. Like I said, please feel free to interrupt us at any time if you have questions with your mic, or you can put it in the chat. We'll do our best to monitor it. So I want to start with going over an overview of what we're going to cover. So we'll cover, like I said, pretty much everything. Disclaimer, we may or may not cover everything that will be on the exam, but anything that Laura has talked about in class is fair game for the exam. We pick topics and we're going to go over topics that we think are important or that you guys mentioned here, but we may not get to everything. So, and like I also said, we will send out the slides with our notes and the answers after the session. So first, some general exam tips. You definitely want to make sure you are reading the full question. So sometimes Laura will put like a big paragraph of information and not all of it will be important, but you want to try to pick out the parts of the question that are the most important and make sure you look at the question and you answer every part of the question. So if you don't answer every part of the question, you might not get full points. So one way to check to like reference how solid your answers are using the problem sets as a reference for the level of detail that you should put for your answers. Make sure you're explaining your answer. Laura said in class today that she'd rather you explain too much than too little, but be careful. Everything you put down has to be correct. So you could end up like not getting full points if you say something that's incorrect when you explain too much. So just be careful. And I know this is kind of obvious, but try your best not to overthink it. These problems are meant to like assess key concepts and details. So use what you've learned. I promise you guys know more than you think you do. So it'll be okay. And also when you're studying the rest of the week, don't focus on memorizing things because remember, this is an open book exam. So these questions aren't going to test memory based. They're going to be more problem solving based. And yeah, so now we get to go into the fun stuff. Let's talk about some chemistry.
Yep. So first we're going to start with, amino acids. This is what we started the semester off with. Basically for this class, you don't need to know the specifics about each amino acid. If you take the MCAT or the DAT or something in the future, you will know all of the specific details of each amino acid. But for right now, just know that all amino acids have an amino group, which is in blue here, and this is labeled also. Carboxyl group, a hydrogen, and the R side chain, which is the variable part of the amino acid. This R group gives each amino acid its unique chemistry. This is what determines whether it's polar, nonpolar, acidic, basic, neutral, whatever. So there are 20 amino acids. 2 are acidic, 3 are basic, 6 are neutral. These are your polars. So when she says polar uncharged, she means these polar neutral amino acids. When she says polar charged, she means either acidic or basic. Yeah, you do need to memorize all of them for the MCAT, but for this class, you do not. At physiological pH, she might ask you about what their structure would be. At pH of 7.4, your acidic amino acids are negative, your basic amino acids are positive. It's the same with the amino group and the carboxyl group. And then another big point to memorize about these amino acids, because she's going to ask a lot about protein folding, is that nonpolar amino acids are hydrophobic and polar amino acids are very hydrophilic. Okay. So peptide bond formation. Your peptide bond formation is through a dehydration reaction, which is this, as you can see right here, it's going to be between your carboxyl group of your first amino acid and your amino group of your second amino acid. You have this OH from your carboxyl group and the hydrogen from the nitrogen group. You lose these as water. That's dehydration. Another name for dehydration is condensation. So if she asks you about condensation, dehydration. This is peptide bond is a covalent bond. It's very strong because you don't want your backbone to be breaking apart. It's catalyzed by the RNA of the ribosome. And remember that amino acids are always read from your N terminus to your C terminus. So that's why the back end of your first amino acid and the front end of your second amino acid are what are making that bond. So if she says the N terminus is read first, or we'll get to that with signal sequences, but your N terminus comes out first, that's what's translated first, that's what you'll get first. And just for reference, when you're breaking a peptide bond, if she ever mentions peptidases or something, that is breaking your amino acid bond, your peptide bond, with an input of water. So you're making a bond by losing water, you break a bond by putting in water. Does anyone have any questions so far? Are you guys good? You can go to the next slide, Jason. Oh, can you talk slower, please? Yeah, sorry. It's just we have a lot to go through, so I'm trying to get through it. But
don't worry
yeah,
too much about writing everything down, because we will send out the slides. So, but yeah, we'll try to talk.
We are recording this too. Yeah, we are.
And it is being recorded.
Yeah. Okay. So with your protein structure levels, we're starting with primary. Your primary protein structure level is the sequence of amino acids held together by covalent bonds called peptide bonds, like we just talked about. So your primary structure is literally just the sequence of amino acids. Your secondary structure is the formation of alpha helices or beta sheets, which is caused by hydrogen bonding between the amino groups and the carboxylic acid groups. This is important for secondary structure, is your R groups are not included in this bonding. They stick out on the sides of the alpha helices, and they're either above or below the planes of the beta sheets. They do not interact at all with this bonding. It's really, she wants you to focus, the hydrogen bonds are a big point of the secondary structure. And the hydrogen bonds are non-covalent bonds. Okay, so just remember that it's the backbone. So the backbone is just your NCC pattern, okay? And then we're gonna do a slide on hydrogen bonds in a second. One second. Okay, sorry about that. So this is a quick note on hydrogen bonds because I know that there's a lot of confusion with hydrogen bonds. So she doesn't use the terms H bond donor or H bond acceptor, so do not worry about the naming at all, okay? It's not necessary for your exam, it's not important. Basically, what you need to know for your hydrogen bond is that it needs to be between a hydrogen on one molecule and a fluorine, oxygen, or nitrogen on the other. Don't worry about fluorine, it's not used in cell biology. We usually don't talk about it. Just know the oxygen and nitrogen. Your one molecule has to have an oxygen or a nitrogen bound to a less electronegative atom. So if you see in this example, you have two water molecules. This one oxygen is bound to a hydrogen. Hydrogen is less electronegative than oxygen. It makes oxygen a negative dipole. On your other molecule, you have your hydrogen, because your hydrogen needs to be bound to either an oxygen or a nitrogen in order for a hydrogen bond to form. So when you hear H bond donor, H bond acceptor, don't think about it because you're not going to hear it. She's not going to mention it. It's just know that your one molecule needs to have that negative dipole, and the hydrogen on the other one needs to have a positive dipole. Does anyone have any questions about hydrogen bonds before we move on? Because they're going to come up a lot. No? Okay, cool. Move on. So then we have our tertiary and quaternary structures. Tertiary is the interaction between the R groups. So this is where your R groups really get to show their true colors, and their chemistry is what determines the tertiary folding. Like we said, we'll talk about it, but like the hydrophobic effect will be more with your hydrophobic amino acids due to their hydrophobic R groups are going to be in the middle, and your hydrophilic amino acids with your hydrophilic R groups will be on the outside. A big part of tertiary structure is disulfide bridges, which you can see in this diagram between the two cysteines.
They are covalent bonds between two cysteine molecules, and they help stabilization a lot in your tertiary structure. And then your quaternary structure is the overall structure of the proteins that made up of multiple polypeptides. So you can see in the first diagram how it's just one long chain of amino acids. That's a singular polypeptide. And then the second diagram, you get multiple polypeptides like hemoglobin, which is 4 subunits. So not all proteins have quaternary structure. Some proteins can just function off of tertiary. But remember that disulfide bridges are only in tertiary and quaternary structures. So if she asked you something about disulfide with secondary or primary, that wouldn't work because they're not involved in those, okay? So the most stable form of tertiary structure, like I said, with the hydrophobic effect is that the polar amino acids are facing the outside and the nonpolar amino acids are facing the middle. So we have a quick question here. You guys can either answer with your mics or put in the chat, whatever, and then we'll go over it. But what effect would a molecule that disrupts hydrogen bonding, such as urea, have on protein structure? Or you can think it too. You don't have to put it in. We'll go over it in a second. Okay. Structure remain intact. Secondary structure. Yep. Yep, you guys are right. So basically, with urea disrupting those hydrogen bonds, you would definitely disrupt your secondary structure and you would unfold those alpha helices and beta pleated sheets. It could also affect tertiary. Only occur. Disulfide bridges only occur in tertiary and quaternary. So they are not in secondary or primary. They will be in tertiary and quaternary.
For quaternary structures, do they use all types of bonding?
They do.
Okay.
Yeah, they do. We kind of go into it, I think, a little bit, but yeah, they use. Sorry, they use most of their like all of the bonds except for peptide bonds because that's technically in primary. Like if that makes sense, because that's like your makeup of the quaternary structure, anyways. So
if it helps, later we have a slide that has like a summary of all the types of bond and like which structures have each bond. So that'll be helpful
we have a bond
later.
summary. So we will go over that. But to answer this question, urea would disrupt secondary structure and it would unfold the alpha helices and beta pleated sheets. It would also affect tertiary and quaternary, but it would not affect primary. So you guys got that right. So you would still have primary structure. Okay. And then we have another question. This time, if you guys want to put your answers in the chat, you can go to the next one, Jace. Yeah. So which of the following may be considered an example of tertiary protein structure? You have van der Waals interactions between two phenylalanine R groups located far apart on a polypeptide, hydrogen bonds between backbone amino and carboxyl groups, covalent disulfide bonds between cysteine residues located far apart on a polypeptide. So what do you guys think? It can be more than one too. It's not one or the other. It can be multiple answers. Got 1 and 3. Another 1 and 3. Another 1 and 3. Another 1 and 3. Yep, you guys got it. It's 1 and 3. Yeah, so they're both examples of tertiary structure. Disulfide bonds, you got it. Those are tertiary. Van der Waals on 2 amino acids located far apart on the same polypeptide, also tertiary. Hydrogen bonds, no, that's secondary. Right, you guys got it. Awesome. So domain versus subunits. I know there are a few questions about this in class. Proteins can be comprised of separate functional parts. So your domain is an independently folded section of a single polypeptide. So your domain would be like in one singular polypeptide, like a tertiary structure, you would have a section that is in question, that's your domain. Yeah, where Jason's pointing, that's your domain. Domains are evolutionarily conserved. And then you have your subunit, which is a single polypeptide of a protein made up of multiple polypeptides. So like hemoglobin, you have 4 separate parts. of your quaternary structure. So there can be domains within subunits, because your subunits are the whole polypeptide that's in question out of multiple. Does that make sense? I don't really know how else to really explain it, but just remember domain is an independently folded section of a single polypeptide, and a subunit is a single polypeptide made up of a protein made up of multiple polypeptides. Can you explain evolutionarily conserved?
Yeah, I got it.
Yeah.
So basically what that means is if you have, so across like different species, you could find a domain that's like, that has like the same structure or the same like set of genes, I guess, or like proteins across different species. So like a human might have the same type of domain on a single gene as like a alligator or something like something
Yeah.
like that. Like that means
Yeah.
that's what that kind of means.
Yeah. So subunits are only present in quaternary structure. Yes, because you need that multiple polypeptide, and you only have that in quaternary structure. Tertiary is your single polypeptide. So, true or false, a protein with one polypeptide can contain multiple domains. Is that true or false? You guys are saying true in the chat. Yep, that's true. Does anyone want to explain why? Or do you guys kind of get it? All right. Yeah, basically one polypeptide can have multiple domains because a domain is an independent folded section of a single polypeptide. So if you have your tertiary structure and say it's one polypeptide and it's folded, say it's like kind of like a slinky kind of, on one section of like the portion that goes up would be the one domain, and the portion goes down could be another domain because it's different folding. Okay.
Or like
Go ahead.
another example is like like a protein that activates like an ion channel. One part of the protein, one domain on the protein might open the channel, another domain on the protein might close the channel or something like that. So like domains have like a independently folded, and they usually have like a specific function.
Yeah. So Lauren, to answer your question, yeah, different domains have different functions. Correct.
Yep.
Right. So next we get our modeling of proteins. Mainly when she asks questions about the modeling of proteins, she wants you to know the pros and the cons of each model. So if she were to ask which model would be best for looking at R groups, you would not want your backbone molecule model because it doesn't show your R groups. So your backbone is just the backbone, it's in the name. You hear backbone, no R groups. Your ribbon model is backbone and secondary structure. So the secondary structure, you get your hydrogen bonds. You'll see these alpha helices, kind of like that ribbon, and then your beta pleated sheets are. Do all polypeptides have subunits? No, because if it's a single polypeptide, it won't have a subunit. If it's a polypeptide made up, if it's a quaternary structure and you have multiple polypeptides, then yes, it will have subunits. But a singular polypeptide will not have subunits. So your ball and stick shows every atom, including R groups. Your space fill model has every atom and the space that it takes up, so that's great for seeing your overall shape. You can also use multiple different models. That's what overlaying different models means. But yeah, if you see a question on the exam about these modeling, it's usually she's like, which one would be best for showing your R groups, or which one would be best for showing secondary structure? And you would say ribbon model for secondary, ball and stick, or space filling for R groups. And speaking of R groups, we get our R group interaction. So here are all your amino acids. You got your polars, your charge. So anything with a charge is a polar. So the positive charge is your basic and your negative charge is your acidic. And then these other polar amino acids are uncharged. And then your nonpolar are also uncharged. So like I said before, your polar molecules are hydrophilic. Your nonpolar molecules are hydrophobic. The hydrophobic effect is basically the nonpolar molecules that kind of want to hide in the middle. H bonds are not considered covalent. H bonds are non-covalent bonds. Yeah, no problem. The hydrophobic effect is your nonpolar molecules want to hide. They don't want to touch the water because they're hydrophobic. Your polar molecules do want to touch the water. They're hydrophilic, especially in aqueous solutions. So the main takeaways from your R groups that you want to know is these polar charged R groups, so your basic and acidic amino acids, will typically make ionic bonds, especially electrostatic interactions. You get that positive charge, you're going to want an electrostatic interaction, which is your very, very strong bonds. Not as strong as covalent, but whatever. Your polar uncharged R groups will typically make your hydrogen bonds because they have those negative and positive dipoles, and you have a lot of oxygens and nitrogens available for hydrogen bonds. Also, that difference in electronegativity is what gives them the negative and positive dipole. And then your nonpolar R groups will make those covalent bonds because they're hydrophobic. Anyone have any questions? Does that make sense? We have the whole bond thing at the end too, bonds, a summary of bonds. So if you get confused, hopefully that will clear things up. So your steric hindrance, if some of you have taken organic chemistry already or in it, you're definitely going to hear this about bulkiness and repulsion. So the R group, but for this class, you kind of need to know that structure equals function. You'll hear Professor Francis say that all the time, and that applies to your amino acids with their R groups. So the R group size can affect the structure of a protein. So the size and the chemistry are important. An example is like well, we have examples here, but just if you think about like a cell membrane, steric hindrance is annoying in orgo. If you think of an aromatic amino acid in the cellular membrane, it's going to be bulky and big. It's not going to fit. So that, you don't have to know that it's class, but it's just an example. So here's some examples below. Glycine is a very small R group. It's literally just a hydrogen. There's little steric strain, and it might be more flexible within the backbone of a polypeptide. Whereas you have proline, which is cyclic. The backbone is kind of intertwined into the R group. If you see this like kind of pentagon shape, the amino group is connected to the R group, which makes it very rigid and makes it much less likely to rotate or be flexible. And proline can form a helix by themselves, mostly because of this R group interaction with the amino group. Proline's the only amino acid that has this, by the way. So it's the only one that has the amino group attached to the R group, which makes it very annoying to deal with. And then phenylalanine is another aromatic example of a amino acid. This means it's very stable because you have this benzene ring. And then so that means high steric strain. And once again, the backbone is more rigid compared to glycine, where it's just the hydrogen. So basically just the main takeaway for this is, if she gives you, because you don't have to memorize structure at all for this class, you do not need to know. You just need to know like their chemistry and like what will happen if they interact. But if she tells you in a question like you have a very large aromatic amino acid, what will that do to the backbone? It will make it more rigid. It will be less flexible. So don't worry too much about structure because she does not expect you to memorize structure at all for your amino acids. Can you talk about the application of the steric strain? Basically, with steric strain, it's kind of just like, it's more like turning, isn't it? It's like around the bonds. I'm sorry, guys. I took the MCAT this summer, and it's literally all fleeting.
Steric strain is like, it's like what happens when big atoms like come too close together.
Yeah, it's like strain on the
repel
bonds
from each other.
Yeah.
So one way that this might come into play is, for example, you see that this phenylalanine has this big benzene ring that might not always fit in certain places. So it has way more steric strain than something like glycine, which only has a hydrogen that can fit in way more places. So that's where that comes into play.
Yeah, it's the repulsion. I don't think you need to know that really for this exam. But if she talked about it in class, it's fair game. So always ask.
Yeah. The next slide We'll have an example question.
Yeah.
Let's go over that.
So would an uncharged polar R group with a non-polar R group form like a dipole-induced dipole bond? I would say yes. If you have uncharged polar R group with a non-polar. Yeah, because you have that dipole on the uncharged polar, and it would induce a dipole on the non-polar.
Yeah, she doesn't use dipole-induced dipole term, I think.
Yeah, she
She
doesn't.
just uses dipole, ion dipole or something.
Yeah.
But
yeah.
I think that is what I think she just calls it ion dipole.
She would just call it ion dipole. So if you said ion dipole, you'd be, I think you'd be correct.
I mean, dipole-induced dipole is technically correct. So, but
Yeah.
I think she just in this class she uses induced dipole.
Like, you wouldn't be marked wrong for using it if you used it correctly. But what aren't the ions with the charged R groups? Yeah. So we're gonna go over that in the bond summary, which is
that's
the next slide after this one.
We'll come back to that.
Yeah. So we'll come back to that because we go literally over all of it. We're just reviewing the properties of R groups to talk about how they interact, right? We want to know. Yeah. So Benedict, yes, you just have to know how they interact because her main thing is you really need to know all this just to know how these proteins work because our biggest thing is structure equals function. You're going to see that a lot throughout this PowerPoint. So yeah.
And yeah, be ready to she'll ask questions where you'll swap out an amino acid for a different one and then what's the function? That's the kind of questions she'll ask for this.
Yeah.
And that's where the chemistry of the R groups comes
right.
into play.
So on the last slide, if she was like, what would happen if you switched a glycine in the backbone with phenylalanine? It'd be much more rigid or something like that because it's much bigger. On this slide, so these are some more examples. So your tyrosine versus phenylalanine. Tyrosine is a polar molecule. Phenylalanine is nonpolar. Tyrosine is also uncharged. And it's polar because it has this extra OH group. Other than that, phenylalanine and tyrosine have the same exact structure. You just have this extra OH, which is what makes it polar. So this is, you would compare if she was like, what if you switched out tyrosine with phenylalanine? Well, tyrosine is hydrophilic, phenylalanine is hydrophobic, so it would change the chemistry of the protein because the hydrophobic amino acids want to be on the inside. And if tyrosine originally was on the outside, that would change the folding and all that. And then cysteine is also important because you need to know that these disulfide bridges between these sulfurs on the cysteine make those covalent bonds in tertiary and quaternary structures. So it's slightly more polar than other nonpolar amino acids, but it's considered nonpolar. So you would see it most likely. She wouldn't try to trick you, but I think you would see that it would be on the inside of the protein. But I don't even know if she'll even ask that. But the main takeaway for cysteine is that they form these disulfide bridges. So Lauren said, would chemistry play more of a role here than size in that example? Yes, for this example between tyrosine and phenylalanine, yes, because tyrosine has that polar. Because she would probably specify in her question that you switch out tyrosine, a polar amino acid, with phenylalanine, a non-polar amino acid. She would specify because she doesn't expect you to know the structure. So if she specified that you switch polar to non-polar, or she would say you have two non-polars, but one's bigger and bulkier than the other. So she would specify because you don't need to know.
Or she might expect you to refer back to this chart, which
she might give you the chart too. Because it technically is open
yeah,
book.
It's also open notes, so you'll have this chart with you, so don't worry about memorizing.
Yeah. Just know that chemistry and size would change because if she asked you a question about that, it would be if you switched one out for the other, what would happen. Sometimes it would be none, nothing happens. You could switch out glycine for alanine, which is just the carbon group instead of a hydrogen, and wouldn't really affect it. So the larger the R group, the more rigid it is. Usually, yes. A lot of times, if they're aromatic, it makes it a lot more rigid. But yeah, in this case, if the larger the R group, yeah, the more rigid and bulkier it would be. It'd be harder to fit in the protein. So, practice question. Where would you find these amino acids in a folded protein? I kind of already answered this. But polar uncharged exterior, nonpolar interior. Yep. That's
So-
that's basically it. We kind of just went over that anyways. But
So tyrosine, where would that go? That would be inside, right? Phenylalanine would be outside because it's nonpolar. Cysteine is a special case.
no, Jason.
Wait, in
You said polar charge would be on the interior and nonpolar
yeah.
on the
So
exterior.
sorry. Polar is the interior, and nonpolar is out exterior. Cysteine is a special case. It's technically nonpolar, but it depends what it's being compared to. So, like for example, tyrosine is more polar than cysteine. So just be careful with cysteine.
It all has
I've
to do
seen
with this electronegativity. So like oxygen is more polar than, is more electronegative than sulfur. So this is kind of where your Gen Chem and Orgo kind of come into play. Like on the slide with the hydrogen bonding, there's a list, if you remember Fonkelbrish, it kind of is, so your fluorine's your most electronegative, then your oxygen, nitrogen, chlorine. It's on one of the slides if you want to look back at it, if you have questions. But yeah, it's the reason tyrosine's more polar than cysteine is because oxygen's more polar than sulfur.
I just have a quick question about cysteine.
Sure.
I know she's mentioned in the past, like, where would you find this in like a healthy helical structure? And I know it doesn't show up, or maybe it's not cysteine. I forget which one. It's a proline. I'm sorry. The one with the small ring, I believe, was on the side before. Like,
Oh,
I know
proline.
yeah, proline.
Mm-hmm.
Like I know she mentioned like where would they be found on alpha helical structure? Like they would only be found on the ends because of their rigidity, correct?
On the alpha helix. Jason, what do you think?
I've never heard that question before.
Yeah. I mean,
don't know.
I can see why your logic would be yes, because it would make the backbone more rigid. But yeah, I would say on the ends. Yeah, we have literally right here, not located in the middle of the polypeptide.
Yeah, there we go.
Okay. Yeah,
Yeah.
it'd be on the ends because it's super
Okay.
rigid.
I just wanted to make sure that that was on the ends if she were to ask it. That
Yeah,
it wouldn't be found commonly in the middle.
Yeah.
Mhm.
Yep.
Okay.
Cool.
Thank
Well,
you.
Thank you.
No problem. And now we have our bond summary. This is kind of everything you really need to know. Your covalent bonds, your peptide bonds, those are literally only in your primary structure. They hold amino acids together. They make your polypeptide. Your disulfide bonds form between cysteines only in tertiary and quaternary. You will not find them in secondary. You will not find them in primary, only tertiary and quaternary. Then you have your non-covalent bonds. You have your ionic bonds, which is polar charge with polar charge. So your ionic bonds will never form between non-polar amino acids because they're not charged. So you wouldn't have that ionic bond. That's what she really wants you to know. Your hydrogen bonds can be polar uncharged. It can be polar charged. It's basically back on that hydrogen bond slide is where we kind of we went over, what can bond with what in terms of hydrogen bonds. Your van der Waal interactions are nonpolar with nonpolar. And then you have your ion dipole, which would be your ion, so positive or negative, with a polar uncharged. So say you had a basic amino acid that was positively charged, bonding with a polar molecule like tyrosine, which is uncharged. That's your ion dipole. And then we have your primary structures, only covalent peptide bonds, only backbone interactions between the carbons and nitrogens in your backbone. Your secondary structure is only non-covalent, specifically hydrogen bonds, and only backbone interactions. Your R groups are not important here. And then your tertiary and quaternary structure, you have both covalent and non-covalent, and both side chain and backbone interactions. So basically with your tertiary and quaternary structures, you pretty much have everything interacting with everything, because that's what makes it fold. Does anyone have any questions before we move on to we have practice questions, and then we're going to be moving on into protein, the unfolding response and all that. So if you have questions about this stuff, ask them, please. If not, we'll go on to questions. All right, so number 1, true or false? In an alpha helix, the R groups of the amino acids in the alpha helix are available to make interactions with other parts of the protein and or with other molecules. Is this true or false? You got a false. have a true, another true. Yeah, it's true. So it's true because your R groups are sticking out, which means they're available to make interactions. They're not in the inside, especially with alpha helices. So yes, this is true. Oh, why? Sorry, didn't read the next question. What is why? Why? B. Yeah, it's B. Okay, cool.
Sorry, I made this question.
No, it's fine. You're fine. Okay. If you mutate your protein of interest at the 21st amino acid, have you mutated an amino acid closest to the N terminus or the C terminus? What do you guys think?
How long
Yeah,
is our chain? Just so we know.
So-
You're not given it.
Exactly. That's a
part
key
of, that's
point
part
here.
of it. You got a lot of A's. You got a lot of A's. Depends how long. Well, you're not given how long. What would you pick if this was on your test?
Laura won't always. Well,
She does
okay, I'll
not always
in a second.
explain in detail. Neither. That's not an answer. A or B. You got a lot of A's. All right, C. C. Okay, so the answer here is the N terminus. It's A, because pretty much all proteins are going to be at least 100 amino acids long. Okay, there's only a few that are really short. So your N terminus, because if you're a 21 out of 100, you're closer to the N terminus. So most of your proteins are at least 100 amino acids long. It's kind of a trick question on this one because, yes, you guys were right. It does depend. If this was a 21st long amino acid, you'd be closer to the C terminus. But if she doesn't give you that, you should assume that it's closer to the N terminus in terms of the number that you're given out of 100. Okay, so if she doesn't give you the specific number, just assume that it's 100 or more, and that will determine your answer. So yeah, that was kind of a
that.
trick question, but because
I
sometimes she
have-
won't say it.
Yeah, I have seen her ask a question where she doesn't do it, and it'll be something like this. Yeah.
Yeah.
Okay, next one.
So Gandhi says, can you explain dipole-induced dipole? So
She doesn't use
dipole, she doesn't use that term, dipole-induced dipole. You know, you hear it a lot in Gen Chem and Orgo and all that crap, but dipole-induced dipole, you just really need to know that you're going to have that dipole from a polar molecule, and it's going to be inducing a dipole, it's in the name, on the molecule it's interacting with. So it can be polar, it can be another, can it be non-polar? Wait, it was in the bond summary. Just know that if you're thinking dipole-induced dipole, it's usually ion dipole. So you just need that positive ion. Or wait, am I saying that backwards, Jason?
Yeah.
Non-polar with uncharged polar. Yeah,
Yeah.
sorry, thank you. I just had a brain fart, but yeah. Don't really worry about dipole-induced dipole because she doesn't use it, so she's not going to ask you dipole-induced dipole. Okay. So more practice questions. 2 different proteins have the same domain in their structure. Which of the following is true? A, both proteins have the same primary structure. B, at least some part or both proteins have a similar structure. C, both proteins have the same primary structure but different function. And D, both proteins have quaternary structure. What do you guys think? Got lots of B's. We have an A. Mostly B's. Yeah, so you guys got it. The answer here is B. Because domains are evolutionarily conserved, so at least some of the proteins will have similar structure. The proteins are not guaranteed and likely don't have the same primary and quaternary structure. So that's why the answer is B. So number 2, true or false? A protein with one polypeptide can contain multiple domains. What do you guys think? True, 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 true. Yep, true. So this is true because proteins can have multiple independently folded functional domains within a singular polypeptide. If we change this to say a protein with one polypeptide can contain multiple subunits, would that change the answer? Yes. Yes, good. Right, because if one polypeptide cannot have subunits. Yeah, it would be false, right? Okay. So no, this is definitely a question that you could totally see her ask. She loves these types of questions. You have a protein that has a mutation that changes an aspartic acid at the 12th amino acid in the polypeptide sequence to a glycine. Does this mutation impact the primary structure? What about the secondary and tertiary structures and quaternary structure? So what would changing an aspartic acid to a glycine do to your folding?
Or just structures in general?
Or structures in general, yeah. So how would it affect each individual structure, basically?
So in case you guys don't have the chart,
Aspartic acid is a acidic
minus,
polar,
yeah,
negatively
negative
charged. Yep.
And glycine is a non-polar.
Non-polar. Glycine has just
Yeah.
is primary and affect the rest of the structures of protein. Go from negative primary. Yeah. So you guys saying that primary is affected and tertiary is affected, you guys are right. So you're changing the polypeptide sequence, so you have a change in primary. So yes, that's correct. It does not affect your secondary structure because your R groups aren't interacting. It doesn't affect. Your tertiary and quaternary structure, definitely tertiary would be affected because this will change how the amino acid folds because you're going from a polar charge to a non-polar, which is not charged. So this would definitely change how your protein folds in on itself. And then quaternary could be affected if the protein has multiple subunits. That would affect quaternary if it has multiple subunits. If it doesn't, then it would just not affect tertiary. It wouldn't affect secondary because you're not changing your backbone, and that's the only interactions in your secondary structure. Won't this R group fold? Secondary structure doesn't fold with your R groups. It's literally only the backbones interacting. So when you get those alpha helice structures, it's literally just the hydrogen bonds of like the carbons and the nitrogens in the backbone just interacting with each other. So the R groups like just are not involved. When you get what you're, so yeah, no problem. So Lauren, what you're saying with the R group folding interior, that would happen in tertiary structure. So that is how it would fold. It would fold in the interior. So yeah, so tertiary structure would change because you're going from a polar charged, negatively charged amino acid to a non-polar amino acid, which is you're going from hydrophilic to hydrophobic. So in your tertiary structure, when you're folding in on it yourself, when your protein's folding, your polypeptide, originally you would, that at the 12th amino acid in your sequence would be usually facing the outside because it was hydrophilic, but now that it's hydrophobic, it would go in the interior. So that's why tertiary structure would be affected. Secondary structure would not be affected because you're not changing anything about the hydrogen bonds. Yeah, Jessica, basically, if you broke those non-covalent hydrogen bonds, you would affect your secondary structure. You also say quaternary if it was affected. Yes, so if you were given, if she added on like, or to answer this question, if you wrote quaternary structure would be affected if the protein had multiple subunits and multiple polypeptides, then yes, quaternary structure would be affected. If you're only given one polypeptide, no, because there is no quaternary structure. Quaternary structure is only when you have more than one polypeptide. But if you do, then yeah, quaternary structure would be affected in this case. Does that make sense?
Why is it that when quaternary has multiple subunits, it would be affected?
Because you're affecting one of the subunits folding. So if you think of it, say you have like hemoglobin, right? You have 4 subunits. Say in one of those 4 subunits, we're doing this mutation to one of the polypeptides that's involved in one of the 4 subunits. So that subunit within hemoglobin would change because its tertiary structure has changed for folding. So it would be like that one section would be folded differently. So as a result, its interactions with the other 3 subunits in the hemoglobin molecule would also be different because it's interacting differently. Because now instead of having an aspartic acid on the outside, you have a glycine on the inside. So that's why it would affect it if it had multiple polypeptides, because you would affect the quaternary structure. Does that make sense to everybody?
Yeah, thank you.
Yeah, no problem.
Any other questions before we move on?
Because now we're going to move on to a different topic.
Where did my job go?
No, we're good. All right. Jason, your turn.
Woohoo. All right. Here's our meme for this section. If you put this on an exam, I will laugh and it'll make my day, but don't actually do that because you won't get any credit.
You can
Okay.
add it on.
Yeah.
Add it at the end of your answer if you want. Make us chuckle.
Put it in quotes that Jason said that. Okay. All right, so we're going to talk about protein unfolding and misfolding. Some things that could make a protein unfold or misfold are environmental factors like temperature, pH, and salt or light could also have effect. For everyone here, here's a Tinder pickup line, guaranteed 100% success rate. I tried it. I think it worked.
Oh my
So
God.
I'll let you know how it goes. Anyway,
Christ.
so cells don't want to have like crazy changes in temperature because that can impact the protein. And the key thing here is these environmental factors affect non-covalent bonds. So covalent bonds wouldn't be affected by this. If a covalent bond is broken, if a covalent bond is broken, the protein is beyond repair anyway, so it wouldn't matter. Misfolded proteins are abnormal proteins or something that is functioning incorrectly. And this is called denaturing when a protein unfolds. When it refolds, it's called renaturing. And this is obviously harmful to the cells. So cells have a mechanism to fix this called unfolded protein response, which we'll get to in a second. And by the way, I'm going to go through this stuff pretty quickly because we're running short on time or we're behind and we got a lot to get through. So cells want to be able to fix this. They could do this by heat shock response or the unfolded protein response. Lots of problems are caused by misfolding proteins. So mad cow disease or prions is caused by misfolded proteins. She didn't talk about prions this year, but she usually does. So don't worry about that. Alzheimer's disease is actually caused by misfolded proteins too. So that's a big one. So the key thing for this slide, unfolded proteins or misfolded proteins aggregate because when they unfold, their hydrophobic portions are exposed. So they want to hide those hydrophobic portions. And they do that by clumping together to hide the hydrophobic portions. When they aggregate, the protein becomes insoluble because the hydrophobic portions are exposed, so that changes the solubility. Rachel, let me know if there's any questions in the chat because I can't see it anymore.
Yeah, you're good. Someone said you're a Chad, though.
I am not a Chad, absolutely
Yeah, you are.
not. You think so?
Yes.
I don't think so.
Okay, well.
Anyway, chaperones are sorry, that threw me out. Chaperones are proteins that help other proteins fold, or they also work by sensing misfolded or unfolded proteins. During the heat shock response, if the cell experiences a rapid change in temperature, then chaperone production will be upregulated to help fix the misfolded proteins. This response occurs in the cytosol and the nucleus. This is the big part for this section, unfolded protein response. So this occurs in the endomembrane system in the ER. Cells sense unfolded proteins in the ER and they respond by doing a couple different things. They reduce the amount of proteins, they make more chaperones, and they make space for more proteins. The mechanisms for doing this, not necessarily in order. So they decrease translation to reduce folding load in the ER, so they'll stop translating new proteins. They'll also upregulate chaperone production, so you can refold. So like I said before, chaperones are important for helping to refold or fold proteins. So you want to have more of them to fix the unfolded proteins. You could also increase lipid biosynthesis. Biosynthesis just means make more of a biological molecule. So you want to, lipid is like the membrane of the ER. So this just means that basically make the ER membrane bigger to make more space in the ER. The ER will also retrotranslate proteins out of the ER to the proteasome, which is in the cytosol. Retrotranslocation just means go backwards. So proteins are initially moved into the ER, so retrotranslocate would mean to move out of the ER. Any questions on that?
There's none in the chat so far. I think you're good. We're talking about me taking the MCAT, so I'm so sorry. Yeah, no, I think you're good.
So practice problem, where in the cell is the unfolded protein response initiated?
Got the ER. Anyone else wanna add? ER?
Yep, that's right.
Yep.
They're made in the ER. Which of the following are ways that cells respond to unfolded or misfolded proteins? So there's some trick questions here, so be careful. So the first one, do cells upregulate translation?
We have B and D. B.
Did I not finish
Jason,
saying
did
the answer key? That's awkward.
I
Okay.
was gonna say,
so
did you put the answers in?
No, that's awkward. Okay. Well, so they would not upregulate translation of other proteins because you don't want more proteins. You would increase chaperone production because
Yep.
you want more chaperones. You would not translocate proteins to the ER. You retro-translocate proteins out of the ER, and you would increase lipid biosynthesis. So yeah, people
Yeah.
say B and D are correct. Okay. And
Puppy break.
here's our first puppy break of the session. I hope you guys enjoy these very cute puppies.
We'll take like a 30-second to a minute break.
Just to catch our breath real quick, and then
Yeah,
we gotta
and
get
look at
going.
these cute puppies because they're really cute.
Okay. What am I
Ugh, okay, I'll watch Pokemon in 30 seconds.
All right, now we get to talk about
Enzyme. Quick Among Us. Yeah, let's all play Among Us. All right.
Right.
So protein ligand interactions and enzymes. Wait, Jason, did you want to
Woohoo.
do part of?
Can you do enzymes? I'll do the first part.
Yeah.
Okay. So a ligand is anything a protein binds to. It doesn't necessarily always have to involve, it doesn't necessarily always have to involve a binding pocket. It could be a flat surface. But the protein does need to be oriented correctly as well as the amino acids exposed in the pocket. I don't know exactly what that means, but the protein just needs to be in the correct orientation for something to bind, and that the orientation matters. Affinity is the strength of the interaction between proteins and the molecules binding to, so strength between protein and molecule and ligand. And we'll talk about KD in a second. Affinity is the number of bonds between protein and ligand, and it's also the strength of those bonds. So, and also a key thing here is that the Goldilocks hypothesis that she kept mentioning, you don't necessarily always want a strong affinity because sometimes you want things to release. So you want something that's like just right for functionality. KD is the dissociation constant. That's key dissociation, not association. So that means that KD has an inverse relationship with affinity. So as you decrease KD, affinity goes up. She won't ask you to do things with this equation really. You just know that this equation is when the concentration of ligand is this equation equals the concentration of ligand when half the binding sites of the protein are filled. Like I said, a smaller protein equals a stronger protein and ligand binding interaction, so harder to release. And yeah, there's an inverse relationship. Any questions about KD? Because this is an important concept. She doesn't usually ask questions directly about KD, but she'll like include it in other concepts that like
You'll never have
involve
to
affinity.
calculate anything either.
Yeah.
So like she would like
Okay.
give you the KD, but yeah,
Yeah.
no, you're good. All right, so enzymes. Enzymes are biological catalysts. They increase the rate of a reaction by lowering the activation energy required for that reaction to occur. You'll see that graph on the next slide. Basically, they do this by stabilizing it. So lower energy equals more stable. They make and break covalent bonds. This means if you're making and breaking covalent bonds, you're changing the chemical makeup and the structure of these molecules. So these are chemical reactions. Enzymes are needed because they speed up reactions that would happen on their own, but are too slow for the function of the cell. An example is when we do nuclear import and export is RAN-GAP. Like she said in class, technically RAN can hydrolyze GTP without GAP, but because it's too slow, the cell like won't function, so it's basically obsolete. So enzymes can be regulated. This means that the cellular processes that rely on enzymes can also be regulated. So if an enzyme is required for a certain function to occur, if the enzymes are regulated, that means cellular processes can also regulate those functions. And a quick thing is not all enzymes are made of 100% protein. Some are made of RNA. These are ribozymes. So I have a question. Is this true? Enzymes are catalysts. All enzymes increase the rate of chemical reaction, change the phase reactions, and the reaction. If not, why?
I
So,
don't know about the last part of that. Like,
oh,
do they always change the free energy?
of the reactants and the reaction.
Or I guess they lower it, yeah.
I think the answer was yes to this question because I remember seeing it. It was false. Oh, okay. It
I
was
think
false
it's
on the
no because
Yeah,
the energy of
they
the
don't change
reactants doesn't change.
Yeah.
Oh, you're right. Yeah, that was
It's
a
a
big
transition
thing.
state.
A big thing to know is that enzymes just change the kinetics, not the thermodynamics. If you really know your chemistry and your biochem, the energy of the free energy is a thermodynamic property, and that's of your reactants and your products. Enzymes do not interact with that at all. They just change the kinetics of the reaction. So that's why it's false on the quiz, because free energy is a thermodynamic property and enzymes only affect kinetic properties. Yeah, no problem. And then, yep, so this is the reaction coordinate, basically. If you see here on the graph, you'll see that products and reactants, their energy is the same with or without the enzyme. So that's kind of just to go off of that. So the transition state is usually very unstable. That's why it's very high in energy. The enzyme stabilizes it, so it lowers the energy. So lower energy equals more stable. Just some terminology. A spontaneous reaction is the energy of the products is less than the reactants, so energy is released. So spontaneous reactions occur. We want spontaneous reactions to occur because you're releasing energy, and increasing the entropy is kind of more the biochem side, but so this is an example of a spontaneous reaction. Another name for spontaneous is exergonic. Exergonic means just the energy is released. And then non-spontaneous reaction is if the products have more energy at the end than the reactants. So if this line of the products was above reactants at the end, that means that energy has been required to put in, and this is an endergonic reaction. So non-spontaneous won't happen on its own. It needs something else. But that's kind of more biochem when you talk about reaction coupling and all that stuff, but you don't need to know that for this class.
She doesn't talk much about the reaction coordinates.
She really doesn't. It's just kind of there for your own visual.
Yeah, the key part is that enzymes lower the activation energy and they help make it faster. And the next slide that we're going to talk about is how they do that.
Yeah. So this is how enzymes catalyze chemical reactions. Enzymes are highly regulated, like we said. That's how cells regulate the processes that enzymes control. You have these.
The question also.
We can skip that question.
The answer will be in the slides posted if you're super curious, but we'll skip it.
Yeah, it's really not necessary. This is actually a question I took from MCAT prep, so it's really not that important. Okay, so you have these three scenarios. A is just when it's encouraging two substrate molecules and orienting them in the right way so that a reaction will occur. B is rearranging the electrons to create partial charges that favor a reaction. And C is the enzyme is straining the substrate to force the transition state to favor the reaction. That's basically it, enzymes.
Cool. Any questions about enzymes before we move on? Okay.
ATP World Tour.
So now we'll go into protein function regulation. This is just really quick. So allosteric regulation is when something binds to a spot other than the active site. So the active site is here, an allosteric site would be somewhere else. She doesn't talk much about it. She doesn't really like allosteric regulation, but the key thing, structure equals function, like we keep saying. Post-translational modifications or modifications that occur after the protein is made. So after tertiary structure, these are reversible. They let you switch mechanisms so you can change the surface chemistry, change structure, which changes function. Some examples are methylation, ubiquitination, acetylation. For example, I don't know if she mentioned it yet, but you've probably heard of it in Bio 151, ubiquitin-mediated proteolysis. So that's when a cell is tagged with the ubiquitin and then it is sent to the proteasome. So that's one way that the cells do that with these post-translational modifications. Nucleotide binding and hydrolysis. So what were we focusing on here, Rachel?
Just that GTPases and ATPases are the categories of enzymes that hydrolyze GTP and ATP respectively. GTPases only hydrolyze GTP, ATPases only hydrolyze ATP. It's never both, it's only one or the other. And their function is to turn a triphosphate to a diphosphate. That's the main thing to take away from the slide is that when you have your GTPase or ATPase bound to the triphosphate, so GTP or ATP, you're on and you're active. And then you have this hydrolysis where you lose an inorganic phosphate and you go from tri to di. And when you're bound to your diphosphate, GDP or ADP, you're off and you're inactive. So this is common in signal transduction pathways, which we'll get to later in the semester, but that's really all you need to know is that GTPases only bind GTP and ATPases are only ATP, and whether they're on or off.
Mm-hmm. Cool. All right, so now my favorite stuff, protein localization and control. I know this picture gets me every time, but okay, so signal sequences. All proteins start translating on a free ribosome in the cytoplasm. If they have another signal sequence, then they can get moved somewhere else. So if the protein has an ER localization sequence, an SRP will drag it to an SRP will drag it to the ER, and then it enters the endomembrane system. Every time a protein is localized to the ER, it must have an ER signal sequence. If it has an ER signal sequence, then it will never. Okay, so let me repeat that. If a protein has an ER signal sequence, then it enters the ER, and then once it enters the ER, it almost never comes out unless it's being exported from the cell. So that means that a protein with an ER localization signal sequence will never have a chance to go to the nucleus or the mitochondria or something like that. Yes, if the protein doesn't have any signal sequence, then it will not leave the cytosol. It will just stay in the cytosol and function there. If a protein has to be secreted outside of the cell, it has to go through the endomembrane system. If you don't remember, endomembrane system is like the ER, Golgi, things like that. That's the whole endomembrane system. Once it enters the endomembrane system, they rarely come out. So here's some trends for different signal sequences. Don't ever memorize the order of signal sequences, and they're often like different, but do know like the trends. So for example, NLS is a stretch of positively charged amino acids, and that's like one of the trends you should know. I have I list them all here. I'm not going to go over them, but you'll have this when you're taking the exam. Yeah, so this chart is super useful. Tiffany posted in the Discord, and I think it's mostly used in Bio 151 classes, but it's super relevant here. So I'm going to be using it when we go over practice problems. Like I said, so anytime, so when you're doing problems like these, use this chart. So like, for example, a protein that has an ER signal sequence, it'll dock to the ER and go through the endomembrane system, which is all of this stuff. And because it has the ER signal sequence, it'll never have a chance to go over in these parts to the nucleus or the mitochondria. So I'll come back to this chart when we're doing the problems. Okay, so any questions on this stuff, signal sequence stuff so far, before I move on to nuclear import export? Okay, so nuclear import and export. Before we start, I want to make sure we're clear on the difference between GEFs and GAPs. So it's really helpful to use the names of these proteins when you're trying to figure out what they do. So GEF is a guanine nucleotide exchange factor, and what it does is, as it says in the name, it exchanges GDP for GTP. It binds RAN, which causes a conformational change, and so GEF will bind RAN, it changes the shape of RAN, and then RAN will lose its affinity for GDP, and that lets GTP diffuse into the binding pocket. Just so you know, GTP is present in much higher concentrations than GDP in the nucleus, so that's why it'll diffuse into the binding pocket once you shovel GDP out.
We also have the diagrams on the next few slides, which will help you like visualize it better, but
yeah.
Just
So RAN bound to GDP is inactive, but becomes active when it binds to RAN GTP. Again, using the names, GAP is a GTPase activating protein. So what it does, it stimulates the intrinsic GTPase activity of RAN. So RAN is a GTPase, and what it does is it hydrolyzes GTP to GDP. And that's done in the cytoplasm. Basically, it's just like we said, enzymes increase the rate of reaction. Otherwise, it's really slow, and it's like not worth the effort if it doesn't have GDP. I mean, yeah, if it doesn't hydrolyze it. Okay, so now talk about nuclear import. So the process in the cytoplasm, a protein with an NLS will bind important. The NLS is what allows important to bind to the cargo protein. So it has to have the NLS to bind the cargo protein. Once important binds to the cargo protein with the NLS, it'll enter the nucleus through the nuclear pore complex. The nuclear pore complex is huge, so proteins can enter this fully folded and have no problems. Once it's in the nucleus, it needs to release the cargo protein. So it does that when RAN GTP binds exportin. RAN GDP cannot bind exportin, so you have to have RAN GTP. Like I said before, RAN GDP is switched to RAN GTP with GEF. So it swaps the GDP out of the binding pocket and allows GTP to diffuse in. So RAN GTP will bind, it binds important, and now it cycles it back out of the nucleus to the cytosol. So now RAN GTP needs to release important, so they can both repeat their cycle. It does that when RAN GTP is hydrolyzed to RAN GDP. The protein that does this is GAP. So it makes RAN GTP, RAN GDP. When that happens, the conformational shift happens and important is released. The question says, does important transform into exportin? No, those are two separate molecules. Yeah, those are different. So, but they're both, we'll talk about nuclear export next. Does anybody have any questions about the nuclear import process? Just remember the functions of GEF and GAP, and that RAN GTP is important. It has to bind before the cargo can be released in the nucleus.
Yeah, the important is this little brown circle.
Yep.
Do we need to know about NTF2?
I don't know what that is, so no.
The answer is no.
Can we talk about if something changed in the system? Yeah, we're gonna go over some practice problems, so that we'll hit that.
And then so important binds to let it out too. No, that's different. That's exportin. So
Yep.
important is only necessary to get the cargo protein into the cell, into the nucleus, sorry. Exportin is what takes it out.
Yep. If a protein needs to get out of the nucleus, it has to have a nuclear export sequence, and exportin has to bind. But we'll talk about that next.
Yeah.
So yeah, someone asked, like, what would happen if GAP was hindered? So if GAP was hindered, then, sorry, let me use the cursor. So if GAP was hindered, then what do you think would happen? So, someone answer, and then I'll go from there.
So I would expect that important wouldn't be able to let go of the RAN GTP complex. And without important being let go from that complex, it's not going to be able to bind to cargo protein and allow it to be released into the nucleus. So you'd have an increased amount of RAN GTP important complex in the cytosol, and eventually like the cell would kind of like shut down.
Yep,
Yeah.
exactly. So if RAN GTP cannot hydrolyze to RAN GDP, it'll never let go of important, or if it does, it'll be super slow, and everything that happens would be non-functional anyway. So if GAP's non-functional, then RAN GTP cannot let go of important, and they'll just be stuck together, and neither of them can repeat their cycle. We don't talk about how RAN GDP gets back into the nucleus, but just know that it does. But if it was bound to important, then it probably wouldn't be able to get back into the nucleus either.
And Heather, to answer your question, important needs to bind to RAN GTP to leave the nucleus to get back out into the cytosol so it can be released and then tag another cargo protein to enter into the nucleus.
Yep. So it can only bind the cargo protein, or wait, never mind, sorry. Yeah, what Rachel just said.
Important can only bring the cargo protein in. It can't bring anything out, and it can't go out on its own, basically. It needs to be bound to RAN GTP.
Cool. Next we'll talk about nuclear export, which is the same thing, just reverse. So does RAN GTP bind exportin, or does it bind
Exportin.
Okay. Yeah. So RAN GTP binds exportin first, and then when RAN GTP binds exportin, then it can bind to the cargo protein that needs to get out of the nucleus. If a protein needs to leave the nucleus, it has to have a nuclear export signal sequence. So this NES. Like we said before, GEF's role here is to make sure that RAN GTP is the one that is like present. So exportin cannot bind to RAN GDP. It has to bind to RAN GTP. So once you have this whole complex, it leaves through the nuclear pore complex, and now it's pretty much the opposite. So now you need to let go of the cargo protein in the cytosol. It does that the same way. So GAP hydrolyzes GTP to GDP, and once that is hydrolyzed, then the cargo protein is released into the cytosol. Again, if GAP was non-functional, then it wouldn't be able to release this cargo protein, and the cargo protein would be non-functional because it's stuck to RAN GTP. Any questions about that? And then once the cargo protein is released, exportin is also released, and exportin can go back into the nucleus to repeat the cycle. Any questions about? Yeah, go.
The main thing to remember, the difference is that the cargo protein for nuclear import is just the cargo protein and important that can take it into the cell. But for exportin, you need that triple complex for the cargo protein to leave the cell. That's like the main difference between the two.
Any questions about these? Otherwise, we can go over some practice questions.
Let's go to the practice questions.
Okay.
Is GEF an import too?
Yep. So they have the same function in import and export. GEF just helps make RAN GDP turn into RAN GTP. They both RAN always has to be bound to GDP in the nucleus to do anything. And
yeah, so
GEF is what allows it to do that.
Yeah, GEF basically turns RAN on to GTP instead of GDP, and GAP turns it off.
Okay, so now some practice questions. Number one, if a protein has both an so we're going back to signal sequence first. If a protein has both an ER localization sequence and a mitochondrial localization sequence, where will the protein end up?
Yes, just put it in the chat. ER.
So I'll go back to the chart. Yep, so everyone's saying ER so far. So like I said, the ER, so if a protein has an ER signal sequence, then it'll immediately get docked to the ER and it'll never have a chance to go to the nucleus or the mitochondria. So then it'll enter through the system. If it doesn't have any retention sequences, then it'll be secreted. But my goal with this question is just to make sure you know that it'll go to the ER first and not the nucleus. Okay, so similar, if a protein has an ER retention sequence, an NLS, and a Golgi localization sequence, where will the protein end up?
We have an NLS, we have a nucleus, more nucleus.
So one ER.
Nucleus.
Yep.
Yep, so the answer is nucleus.
Yep, going back to the chart, ER retention is different from ER localization. So ER retention is if you want to get stuck in the ER lumen or the ER membrane. So since it doesn't have the ER localization sequence, it'll be translated by a free ribosome. It'll go through the nuclear import cycle, and then it'll end up in the nucleus. Okay, if a protein has an ER localization sequence, where will the protein end up?
We have a secreted outside the cell. Out of the cell, yep, it would be secreted.
Yep. The first Anastasia's question, wouldn't it need an ER sequence to get to the Golgi though? So it'll never go to the Golgi because it doesn't have an ER localization sequence. If it does need to go to the Golgi, yes, it would need an ER localization sequence first, and then it could go to the Golgi through, and if it had like a Golgi retention sequence or a Golgi localization sequence.
Yeah, don't assume if it doesn't say, like in this question, it just says ER retention sequence. Since it doesn't say localization sequence, you can't assume that it has one.
Yep, so back to number 3, if a protein has just an ER localization sequence, where will the protein end up? So if it just has an ER localization sequence and no retention sequences, then it'll go all the way through this chart, and if it has no other signals, then the protein is secreted. So you guys were right there. Follow the chart. Okay. So these are back to the nuclear import export mostly. So true or false, the protein enters the nucleus fully folded.
Lots of
Great.
trues.
Lots of trues. Okay, next one. True or false, the signal sequence is cleaved off once it enters the nucleus.
Real fast, what's the answer oh wait, never mind, I got it. Never mind, sorry.
Okay, no, you're good.
Okay, we'll also send out all the answers too after if you want to check. Yeah, I saw lots of falses. So the signal sequence is not cleaved off once it enters the nucleus. Follow-up question: which areas, which part of the cells do have the signal sequence cleaved off?
ER mitochondria, ER. Yep.
Yep.
ER
exactly.
and mitochondria, both.
Yep. So most people just said the ER. Remember, the mitochondria also cleaves off the signal sequence once it enters. There was a question, can you go over what co-translation translocation is? So yeah, so going back to this chart, once co-translation just means that it is translocated into the ER at the same time as translation. So they're co-translated and translocated. So once the protein has an ER signal sequence, it'll immediately dock the ER, and as it's being translated, it's immediately co-translocated into the ER at the same time. So that's important because it'll never have a chance to go to the nucleus if it has an ER localization sequence, because it's immediately being put in the ER. Okay, next question. The protein, true or false, the protein enters the mitochondria fully folded. Yep, that's false. Proteins cannot fit if they're folded. They won't be able to enter the mitochondria, so they have to be unfolded, and they will fold inside by chaperone proteins. If you're confused about that, there's a video that Laura posted in the videos folder that explains that pretty well.
Where else does a protein not enter fully folded? ER. Yep, right. The ER. Same thing happens. It unfolds and then gets refolded once it's inside by chaperone proteins.
Perfect. Nucleus is post-translational, right? What? Could you explain what you mean by that? Like, do you mean that an NLS is added post-translationally? I don't think so. I think it's
No.
part. I think it's part of like the amino acid sequence.
Yeah.
So, like I said before, it's like a stretch of positively charged amino acids in the sequence that code for an NLS. Okay, number 7. Where will a protein with no localization sequence end up?
The cytosol, cytosol, cytosol.
Yep,
You guys got it.
It stays on the cytosol. Awesome.
Now we're on
Oh.
to gene expression.
Ferris, cat break real quick. These are my cats. This is my cat. She's very cute. I appreciate her. I'm gonna
She's
take
very cute.
10-second break, and then Rachel will talk about gene expression.
Oh, okay. I try not to sound that upset about it. I'm not upset about it. We like gene expression regulation. We like it.
It's very important.
It is very important. What? Wait, what?
You had like an animation.
Wait, what?
Yeah.
That's what that meant? I tried to delete it. I think I put that in by accident. Oops. Okay, whatever. Maybe. Great. So gene expression regulation in eukaryotes, basically you just need to know that transcription is the main site of regulation of gene expression in both eukaryotes and prokaryotes. It's your main site, it's where you're going to regulate. Transcription factors regulate RNA polymerase binding to promoter DNA. We're going to go more in depth with that with the trip operon and all that. In eukaryotes, chromatin packaging determines which genes can be expressed. You can package your DNA in either heterochromatin or euchromatin. If you're packaged in heterochromatin, it's more densely packaged and the genes are less likely to be exposed, which means they're less likely to be translated. Whereas with euchromatin, you're loosely packed, the genes are more likely to be exposed and they can be translated. So RNA in eukaryotes has to be processed and made stable in order to leave the nucleus so it doesn't get degraded or chewed away by nucleuses. Because remember, with eukaryotes, transcription occurs in the nucleus and translation occurs in the cytosol. So once you make your transcript and your mRNA, you have to move out into the cytosol for translation to occur because that's where the ribosomes are. So RNA is made stable through the 5' cap and your poly A tail addition. So your 5' cap is covalently added to the 5' end, so the first end of every RNA molecule. This prevents the RNA molecule from being chewed by nucleases, so it protects it. Nucleases are basically just enzymes that break down nucleotides. And then your poly A tail is added at the end, and this protects the mRNA from being degraded. So the poly A tail is required for export from the nucleus. Yeah, so that's basically it for this slide.
Just know that 5' cap and poly A tail are super important for like stabilization, and yeah, that's the biggest takeaway.
Yeah. Yep. So RNA splicing, I'm going to go over this pretty quickly because she doesn't really go that much into detail. Basically, in your RNA, or in your DNA, you have, your RNA. I'm so sorry, my brain's not working. So you have your exons and your introns and your RNA, your mRNA that's made from, technically it's not mRNA, it's called HNRNA, but I don't think you really need to know that. But basically, once you get your transcript, so you have here with your exons and your introns, this is after transcription. Introns are transcribed, but they're not translated, so they get cut. Basically, that's what splicing means, they get cut out. Spliceosome is what cuts them out. Once you have just your exons, no introns, that's your mature mRNA, and this is what gets translated. This is called alternative splicing. This allows more diversity of protein products out of a single gene. Oh, one more other thing is that introns and exons are unique to eukaryotes. They are not in prokaryotes. So we have this little chart of eukaryotes and prokaryotes. A big thing that she likes you to know is that in eukaryotes, mRNA is monocistronic, which means you get one gene, you get one protein. That's it. So one mRNA will not code for multiple proteins. But in prokaryotes, it can do that, and that's called polycistronic. You get multiple proteins from one mRNA. I'm not going to go through all of this, but basically, just real quick, your eukaryotes, the main differences is that your eukaryotes package into chromatin, prokaryotes don't. Eukaryotes have to move your mRNA after it's transcribed into the cytosol to be translated. Prokaryotes don't. There's no nucleus, no nuclear export. We're going to go more over basal transcription factors and sigma factors in the next few slides. Just remember, sigma is prokaryote. You don't get sigma in, wait, can you go back, Jason? Very quick second, thanks. You don't get that in eukaryotes. And then these are all the same. They both regulate mRNA degradation, mRNA localization, rate of translation, and protein activity. Okay, can you go? Sorry. So yeah, this is just more in detail about transcription. So transcription factor mediated transcriptional regulation. It's a mouthful. Basically, it's just that you have transcription factors which regulate transcription. So why do we need transcription factors? Because RNA polymerase can't bind to the DNA on its own. If there's no transcription factor, it won't bind. And without RNA polymerase, transcription won't even start. So you have your general transcription factors, which aren't specific, and your regulatory transcription factors, which are gene specific. Sigma factors, prokaryotes, basal transcription factors, eukaryotes. So Zawa splicing happens in eukaryotes because they don't have polycistronic mRNA. I don't know if it relates to whether or not they have polycistronic mRNA or not. I don't think it really matters. I think it's just the fact that mRNA is not mature when it's transcribed because it needs to be regulated more because it's being moved from the nucleus to the cytosol. I guess you could say evolutionarily, yeah, that would make sense. That's why splicing happens, because it gives it more diversity because they're not polycistronic, but I don't know if it has anything really to do with that, but yeah. And then you have your positive transcription factors, which are your positive regulators, which enhance transcription when they're bound to DNA, and your negative suppresses transcription. Your RNA polymerase plus the sigma factor is what you need for initiation of transcription in prokaryotes. This diagram is really good for showing it. This yellow dot is the sigma factor. Yeah, that's basically it. You need this complex to start before you start transcription, and that's how it regulates it. So we have a few practice questions. So why can prokaryotes simultaneously transcribe and translate the mRNA, but eukaryotes cannot? What do you guys think? Got no nucleus. Because of the nucleus. Yeah, so yeah, it's because of the nucleus. Eukaryotes, we have nuclei, which means transcription occurs there, and then your mRNA has to move. Prokaryotes don't have any membrane-bound organelles, so transcription and translation occur simultaneously. Basically, in prokaryotes, when your mRNA transcript is being transcribed, as soon as it comes out of the RNA polymerase, it immediately goes into being translated. So why is mRNA in prokaryotes polycistronic? I don't really know. That's just how its genetic makeup is. It's polycistronic. It makes more proteins, I guess. So true or false? RNA polymerase can bind to DNA on its own. Lots of falses. Yeah, that's false. RNA polymerase needs transcription factors to bind in both prokaryotes and eukaryotes. So 3, which of the following is not true regarding gene expression in prokaryotes? A, prokaryotes regulate mRNA localization. B, prokaryotes regulate mRNA degradation. C, prokaryotes package their DNA into chromatin. And D, prokaryotes regulate rate of translation. Got lots of C's. Yep. You guys are right. Prokaryotes do A, B, and D. They do all of those to regulate their gene expression. They do not package their DNA into chromatin. That is only eukaryotes. I'm just going to name some examples of post-translational modifications for you guys. We went into a few, but basically ubiquitination, phosphorylation, acetylation, protein degradation, and dephosphorylation are all examples. Those are in the notes too of the slides, so we'll send them out. And quickly, last one is alternative splicing, an example of post-translational modification. Gotta know. Do we need to know how those work? No, you don't need to know how those work, Lindsay. We got lots of no's. Yeah, no, it's not an example of post-translational modification. Alternative splicing happens after transcription and before translation. All right, good job, guys. Now we get the fun one. Everyone's favorite, trip operon and RNA interference.
Fun stuff. We even have a special guest lecturer coming in a second to help. I know you guys all love him. So
You love him, you know him,
this,
it's someone you know.
puppy break. This is a service dog in training. It's actually my teacher's pet service dog that she's training. But hopefully I'll get one like this soon. So
it's our high school teacher's pet.
Oh yeah.
We both are from the same high school. Okay. Yeah, he's very cute. But let's talk about operons. So basically with your operon, each operon contains regulatory DNA sequences that acts as binding sites for regulatory, what?
We're almost there.
Oh, Chuggin's passion. For regulatory proteins. You're really gonna find these in your prokaryotes. Basically you have your promoter. This is where your RNA polymerase binds. And the operator is where the repressors bind. The operator is part of the promoter. So the promoter is the main regulatory thing, basically, of these operons. Activators will bind to a sequence of DNA somewhere upstream. So upstream is before the promoter. So this little P section upstream is before that. And those recruit RNA polymerase. So operons can be inducible or repressible. Inducible basically means the operon is usually off, but can be turned on in the presence of a specific molecule. I'm sure you guys all know what the lac operon is. You probably learned about it in Bio 151. Lac operon is inducible, so it's off, and then when you have lactose around, it's turned on. Repressible is what the trip operon is. It's usually on, but can be turned off in the presence of a specific molecule. So we're going to get into details, but in the presence of tryptophan, it's turned off. Whereas in the lac operon, in the presence of lactose, it's turned on. And then these are just some diagrams that you can see if you have a repressor bound to the operator, RNA polymerase can't bind, no transcription, no mRNA made. Whereas the other way, if you have an activator that recruits the RNA polymerase, then you get lots of transcription. So the trip operon, real quick, trip means tryptophan. Tryptophan is an amino acid necessary for protein synthesis. So basically with low levels of tryptophan, tryptophan genes are on. Which means that because they need to make tryptophan, basically. When you have high levels of tryptophan, you're going to turn the genes off because you don't want to make more of something you already have. It's a waste of energy, which is basically why control matters in this next part, because you don't want to waste the bacteria's resources when it has access to them already. And then real quick, is the TRPR repressor protein a negative or a positive regulatory transcription factor? So remember, positive and enhances transcription, negative represses it. Got some negatives. Yep, it's negative. It's negative because it suppresses transcription when it is bound to the operator region of the promoter. That's why. So negative represses, positive enhances. The next one. This is the trip operon structure. You have your P and your O site. This is the TRPR we just talked about. It's a negative, it's negative, so when it binds, it suppresses transcription. When you have no tryptophan, you have no repression so there's no binding to the operator. This is a very simplified version of the attenuator. We're going to get in more detail about it in a second. But basically, this TRPE, TRPD, TRPC, TRPE and A, these are all the genes that code for the enzymes that will make the tryptophan. So this directly from transcription does not make tryptophan immediately. It basically codes for the enzymes that will make the tryptophan. And then this is that leading sequence before these genes are transcribed, which is where we get attenuation and all that, which we're going to get into in a second. And then you have your promoter and your operator. So the TRPR does not bind to the purple region. What do you mean, TRPR? This is TRPR. It doesn't bind to anything. It's just kind of, it's just a negative enhancer.
Wait, sorry, just to clarify. Hi, everyone.
Yeah.
Purple
Sebastian's here.
Hey. Okay, so to answer the question, The purple region is just the gene that codes for the protein, that codes for that R, this thing.
Sorry.
Oh, sorry.
Yeah, you can do it.
Okay, sweet. So this is just the gene that codes for the repressor, but the repressor binds to the operator. That's like what the operator region is for. It binds to the operator and it blocks sigma 70. And remember, sigma 70 is your general transcription factor that helps the polymerase bind onto the operon. So if your operator is occupied by this giant repressor, sigma 70 can't bind onto the operon. If sigma 70 can't bind, then certainly your RNA polymerase can't bind either. Does that answer your question, Lauren?
Yes, in all caps.
Very excited. All right.
Sebastian's gonna explain this all to you guys because when he explains it better, people listen. So we're gonna let him take over for these next few slides because he's very good at it.
All right. Okay. So when I think about attenuation, when I think about everything in the trip operon, it really helps me to consider two different kinds of concentrations. The tryptophan concentration, trip concentration, like what Rachel was talking about, and then there's tRNA tryptophan. So please consider these as like two different things. So when tryptophan is low, like how Rachel was saying, when tryptophan is low, your repressor is not bound to DNA, so your polymerase binds. When tryptophan is high, your repressor is bound to DNA and you can't transcribe anything. So, okay, we know the tryptophan is low, but now let's talk about when tRNA tryptophan is high. Remember, although these go hand in hand, please try to consider them as two different things. So when tRNA tryptophan is in high concentration, check out these codons. Or actually, before I get to that, let's just talk about the polymerase. So tryptophan is low, the polymerase binds, and the polymerase starts transcribing. It transcribes all of this, it goes la 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 until it gets to right here. Once it gets to this region at transcription pause, the polymerase is going to pause, right? It transcribes through all of this like it's no problem, and then it pauses right here. Now, it pauses there so that the ribosome can catch up. The ribosome has to put on the transfer RNA. It has to put on the codons or the anticodons. So now that your polymerase is stuck here, your ribosome is going to start translating. It's going to translate, whatever, until it gets to here. This is important. Mind you, this is around region 1, right? So when it gets to the tryptophan codons, it has to ask itself, do I have the necessary codons to translate this? The answer is yes. In this situation, yes. tRNA tryptophan is high, so it does have the necessary codons to add to here. So it could translate straight through this region with no problem. Again, because there's high concentration of tRNA tryptophan. So it translates straight through this, no problem, and then it stops at the stop codon, which is in region 2. So now that it's stopped at region 2, it's stuck here. The ribosome, it's a bunky protein, and it's just sitting here. Once it gets to region 2, once it gets to the stop codon, the RNA polymerase is going to continue transcribing. It's going to continue making this part, and mind you, once it makes region 3 and 4, 3 and 4 are going to form a hairpin loop. Region 3 cannot form a hairpin loop with 2 because 2 is occupied. There's a giant ribosome sitting right here in region 2. So there's no way for region 2 to bind to region 3. So the only thing that region 3 could bind to is region 4. And what you get is a hairpin loop that looks like this. And when 3 and 4 bind, it like yanks the transcript out, it yanks the transcript out from the RNA polymerase, and the polymerase has nothing, it takes out the DNA. Is it the DNA?
It just ranks out the mRNA, and then
yes,
it can't be
okay.
transcribed.
Yes, someone just answered. Thank you so much. It yanks out the mRNA, so it can't transcribe anymore. So again, this only happens when you have high tryptophan. Thank you. When you have high tRNA tryptophan. When you have, just to reiterate everything, when you have high tRNA tryptophan, it's able to translate straight through this, and then the ribosome stops at the stop codon, which is region 2. Okay, so in the instance where you have low tRNA tryptophan, everything is still the same. The RNA polymerase keeps transcribing. It goes la 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 until it gets to right here. The polymerase stops here to allow the ribosome to catch up. Now that the ribosome is catching up, it's translating, but now it's reached the tryptophan codon region. And in this situation, tRNA tryptophan is low. So it has nothing to translate here. It has no codons to add here. So instead of it stopping here in the stop codon, which is in region 2, it's now stopping here at the trip codons in region 1. So when the ribosome stops in region 1 and the polymerase continues transcribing, it transcribes 2, obviously, and then 3 and 4. And because there's nothing sitting on 2, because the ribosome is sitting on 1 and it's not sitting on 2 anymore, 2 is allowed to hairpin with region 3. And when you get a conformation that looks like this, as soon as the cell gets enough tRNA tryptophan, the ribosome is going to be able to continue translating. It's going to continue translating, it's going to break this hairpin loop, and it's going to make the full mRNA transcript. Or sorry, the full protein. Or whatever. Okay, so does that make sense? Do you guys have any questions about that, OGs?
If no questions, we have some practice problems we can go through.
All right, see ya.
Bye. All right, so what happens when there's very high concentration of tryptophan? Yeah, that's why we brought Sebastian. We brought in the big guns for this. So what happens when there's a high concentration of tryptophan? Anybody have any guesses? High concentration of tryptophan.
You're gonna have like attenuation happen.
No, attenuation will not happen. So what happens is you have to think of it as different situations. So if there's a high concentration of tryptophan, then it'll never go into the attenuation process. The repressor will bind, and nothing will be transcribed in the first place.
Oh, so it's only when there's like medium concentration.
Yeah, exactly.
Okay.
Any questions about that? Okay. All right. Here's a situation that's similar to something Laura might ask. If region 4 is deleted, what will be the size of the mRNA transcript and why? See one full size. Any other guesses? Yep, mostly full size. So what happens is if there's no region 4, then the attenuation mechanism can't, like this 3-4 hairpin can't form, and it won't be able to terminate transcription. So what will happen is it'll just keep going through, and it'll make a full size mRNA transcript. Okay, so this is kind of tricky, so pay attention here. If the trip sequence UGG is mutated to the cysteine sequence UGU, and there's plenty of trip tRNA cysteine around, what will be the size of the mRNA and why? See a couple attenuation, smaller size. Yep. So that's true. So attenuation will happen. Basically, what happens is tRNA cis just acts the same as tRNA trip would in the unmutated version. So what happens is it'll just go through this process and it won't terminate early. Someone asked, can you explain why again for number 2? So if region 4 is deleted, then you cannot form this hairpin because this hairpin is when 3 and 4 bind. And if there's no 4, then this hairpin can't form at all. And this hairpin is what causes termination of transcription. So if you cannot form this hairpin, then transcription won't be terminated and it'll be a full-length mRNA. Does that answer your question? Perfect.
Could you go over 3 one more time?
Yep. So basically, think of this mutation as changing. It just swaps out tryptophan for cysteine. and the mechanism is the same, except it's just cysteine. And if there's, in the situation when there's plenty of tRNA cysteine around, then no attenuation occurs because of like the pauses that Sebastian talked about. And that means there's no termination and the full length mRNA transcript will be made. Does that make sense?
Yeah, thank you.
Okay. This one's tricky. True or false, attenuation will only occur when TRP levels are high. See,
False.
everyone's already been tripped over this question in the past. Yes, it is false.
It is false.
Rachel, can you explain that one?
Yeah. So basically, if there's high levels of tryptophan, the the RNA polymerase will never bind because you're repressing it anyways because TRP levels are high. So attenuation only occurs when you start with low levels of tryptophan and like halfway through the cell's like, oh wait, I have too much tryptophan, I don't need to do this, and that's why attenuation happens because then suddenly you're like, oh, just kidding, like I do have enough tryptophan, I don't need to make more. But if you start high, you're never going to start it in the first place, so attenuation won't happen. Does that make sense? I know that one can be kind of tricky.
Yeah, that was a question on the quiz, I think.
Is that also like the first one because we have high levels of tryptophan? The TRP repressor is just automatically going to bind, so it's not going to get that chance to attenuate.
Yeah, exactly.
Yes, yeah. It literally, like, it won't even, it's literally like that thing where it's like, I won't even give you the chance, you know? It's like, you just can't do it. It just won't happen. So what about medium levels? So I got a few questions about medium levels. That's kind of just that intermediate. So it's like it's hard to determine like where the line is for the cell of like what's enough tryptophan and what's not enough. Basically, I think she would specify and say that there was like plentiful amounts of tryptophan. Like if you start low and then end up having high amounts, that's when attenuation would happen. But I don't really know what the threshold is between where like what's enough and what's not enough. But I feel like she would have to specify because there's really no way to tell from like a question unless you specify, like unless she literally says like TRP levels are high at the start, attenuation won't even happen because it won't even be given a chance to happen. Whereas she could say TRP levels are very low, then you'll know that transcription will occur. But if halfway through you have enough transcription, then attenuation will happen because you don't want to make more. But if you still don't have enough, then the full transcript will be made. So in terms of medium levels, you should really just know that it depends on like what is needed for the cell at that time.
You can also think of it as kind of like a gradient. So like in a real life cell, it's never going to go like directly from like high levels to low levels. So there's going to be some intermediate in between. So like it's also possible that there'll be like some variation in the size of TRP levels. Like some might be small, some might be large, but that's like, I don't think she'll get that into detail.
Go back to the question of enzyme catalyzed chemical reactions slide question.
We didn't go over that question, and we're not going to because it's just like an MCAT question. It's like
no, literally it was an MCAT question. I don't think she'll ask you it.
But if you want to know the answer, we're gonna send out. There's an explanation on the answer key we're gonna send out after. So
yeah,
if you're interested.
What would happen if regions 1 and/or 2 were deleted?
You tell me. What do you think would happen?
Yeah. So we got full size question mark.
Remember where the pauses happen. Yeah.
Yep.
The RNA polymerase wouldn't pause. What do you think the effect of that would be?
RNA polymerase would not pause. So what happens if the RNA polymerase doesn't pause?
It continues going like normal.
Yep.
So that would mean it's still full size, right?
Yeah.
I think so.
Okay, thank you.
Yeah.
Yep. So then it would go into like this mechanism where, yeah, it would, if there was enough, if there was low levels of tRNA, then it'll pause here, and then 2 and 3 will form, and there'll be no termination. Yep, we're about to.
I have a question.
RNA.
Yeah, what's up?
So the question was like asking about regions 1 and 2 were deleted.
Mhm.
So like, why is it that the hairpin between 3 and 4 couldn't form, and then like attenuation couldn't happen? Like, I don't really understand that part. Like for the question where it said like, what happens if regions 1 and 2 were deleted? And then you guys said it would be a full size.
Can you explain that, Rachel?
I'm sorry. Wait, so what was the rest of your question? So if 1
Like,
and 2,
You guys said that the full size would form if 1 and 2 regions were like mutated or deleted or whatever. But like, couldn't the hairpin still form between 3 and 4? That would cause the strand to be.
yeah, but it would depend on how much trip is around. So like 3 and 4 would form if you're like, oh shit, like there's too much tryptophan, and then you would want to stop because you wouldn't want to make that full transcript. But if 1 and 2 aren't there, it just doesn't slow down the polymerase really. It doesn't really affect the length unless you have enough tryptophan. So if you have too much tryptophan, then yes, then 3 and 4 will form. But if you have if you don't have 1 and 2, I mean, there's really nothing stopping it. So it'll just keep going unless there's enough tryptophan already, then you won't need to make more. Does that make sense? Because 3 and 4 are your stop sign. So that's basically your stop sign. That's where you stop making the transcript.
Word.
Okay. Any other questions about trip operon? Otherwise, we're gonna cover RNA interference, and then we're done. So, and then we can also stick around for a few minutes after if anyone has any lingering questions about anything.
Yeah. But we should get to the RNA interference. It's a big one.
So by not stopping at 1 and 2, 3 and 4 can't be made. Yes, I'm pretty sure that's what happened. Okay, so I'm gonna go into RNA interference for time's sake. So RNA interference is post-transcriptional control of the mRNA. So the reason RNA interference exists is it likely evolved to protect cells from endogenous and exogenous harm. So it protects from viruses mostly. So miRNA is endogenously expressed, so our cells like make it. They're encoded within the genome, transcribed by the cell. siRNA are exogenously produced, and they're incorporated, which means that they are brought into inside the cell from something else. So either we could use it for research purposes to knock down a protein, or viruses could inject siRNA into the cell. So she might ask a question about the relevance of miRNA and siRNA to research, because it's kind of pretty important. It won a Nobel Prize very quickly because it was so widely adopted. The way they use it is you can use it as a protein knockdown mechanism, so you can stop a protein from being expressed. And once the protein isn't expressed, then you kind of just see what happens. So you break something, and then you see what happens when you break something, and then you can get at function by doing that. So can anyone think of any problems that may arise when you try to apply these techniques? Because that's another question that she might ask, like what are some potential issues with applying mRNA or siRNA to research or as like a treatment medicine? Because theoretically you could use siRNA as a treatment for a protein disease. So what are some problems that you can think of with applying RNAi? Yeah, I'm seeing a bunch of good hypotheses. So some people said that immune system will attack it. That's definitely possible if you're trying to use it for medical purposes. It might never get to the right place. So you have to be able to localize it to the right kind of cells. and it has to get there safely. So yeah, that's definitely another thing. It could get degraded in the way. So it's definitely not easy. It's definitely very good in a research setting because you can control the factors and you can get it exactly where you want it. But it's much harder to use in a medical setting. And like Laura talked about, they've run into a bunch of roadblocks when trying to apply it. Okay. So the pathway for how these different works, the pathway for miRNA and siRNA are very similar. Like the main difference is that miRNA starts in the nucleus and siRNA starts in the cytoplasm. Okay, so for miRNA, these organisms will, like I said, they will encode miRNA in their genome. The initial sequence they create is called a PRI-MR. This is very much a tongue twister. So the first one is called a PRI-miRNA. After that is made, it is processed by Drosha in the nucleus. Drosha turns PRI-miRNA into PRE-miRNA. And then once that happens, then it can be exported into the cytoplasm. So once Drosha processes it into pre-miRNA, then it can be exported to the cytoplasm. Once it's in the cytoplasm, miRNA can be processed by this thing called dicer. What dicer does is it cuts the RNA, it's an RNA nucleus, so it cuts RNA into segments around 20 to 25 base pairs long. And once it's in those smaller segments, then dicer can bind to, not dicer, the processed RNA can bind to RISC. So when it binds to RISC, RISC takes this is a double-stranded mRNA. RISC takes one of the strands of the mRNA and uses it as a guide strand. The other strand is just deleted. It's degraded, So once the miRNA and mRNA complex forms, then it can try to match to, once the miRNA and dicer complex forms, then it can try to match to the target mRNA that you want to disrupt. So there's a difference between an exact match and a partial match. In humans and miRNA in general, the exact match is much less common. We'll hypothesize about why in a minute. But when an exact match happens, which is very rare, this thing called Argonaute, which is the catalytic component of RISC, Argonaute will cleave the mRNA and the mRNA will be degraded. So when there's an exact match, the mRNA is completely degraded. If there's a partial match, which is much more common for the miRNA pathway, it suppresses translation and the mRNA is sequestered into these things called P-bodies. And there it can either be degraded or stored for later use. Does anybody have any questions about the miRNA pathway so far? I can go over any part of it. I know it's there's a lot of terminology, but luckily you have open notes, so you don't have to memorize all of it. Okay, feel free to keep asking questions. I'm going to move on to siRNA. So siRNA is the same exact pathway, except it starts once it gets to the cytoplasm. So there's no Drosha involved with siRNA. siRNA typically comes from exogenous sources, so it's pretty much always going to be an exact match. So that means it'll be sequestered to P-bodies most of the time, and translation will be suppressed. So like I said, it's the same as the miRNA pathway. Once it's in the cytoplasm, dicer cuts it up into these smaller base pair segments. RISC binds to one of the strands of the double-stranded mRNA, and then from there it can exactly match to an mRNA that's floating around in the cytoplasm. And once it does that, it can sequester it to P-body and silence the gene. Any questions about this so far? A key thing here is that this is a catalytic mechanism, which means that RISC can use one siRNA to cleave a ton of target mRNAs. this is important. I can definitely imagine her asking about why RISC can target tons of mRNA. And the reason is because it's catalytic, so that there's an enzyme, and it can keep matching to whatever mRNAs it finds with the exact sequence match. David asks, siRNA and mRNA are both single-stranded at the end of the pathway. Yes, it breaks the double-stranded RNA and it takes one of them as a guide strand. The other one is degraded always. Sorry. I think I said that wrong. siRNA always cleaves, yes. So it doesn't result in sequestering to P-bodies. That's only for partial match, I think. Did I put that wrong in the other? Yeah. Oh, never mind. I did put right. Yeah. So for siRNA, it cleaves always, pretty much, because it's the exact match. Can you explain why siRNA guide strands can cleave many target mRNAs again? Sure. Because it's not like a one and done thing. So once it has this guide strand, it can keep looking for other mRNAs that have the same target sequence. So it could target one, and then it'll cleave that mRNA. And then if it finds another mRNA with the same target sequence, then it'll also cleave that one. So it can just keep going as much as it wants. So it can target a ton of mRNAs. It doesn't like break after this complex. The RISC complex doesn't break after one use because it's catalytic. mRNA itself isn't catalytic. The Argonaut in RISC is the catalytic component of RISC. To answer Lauren's question. Is that because it is an exact match? When it is an exact match, it cleaves. It doesn't necessarily target lots of things because it's an exact match. But
can this happen then for miRNA if it doesn't matter if it's an exact match, since Argonaut is still used?
Yeah, I think it can happen for miRNA too. But yes, I'm pretty sure it happens for both. But remember that if it's a partial match, then it doesn't necessarily cleave; it'll sequester. Yes. I'm actually not 100% sure on that, but I'm pretty sure it could happen in both. But I would honestly email Laura because I'm not 100% sure. By cleaving, do you mean slicing? Pretty much, yeah. It means just like, sorry, it means just kind of like breaking the mRNA in half. So it'll pick a target in the middle and just like break it. P-body. So P-bodies are, you don't have to know exactly what it is. It's just like this place in the cell where like where RNA will get sequestered to. And when they're in the P-bodies, they can either be stored for later use or degraded there. Off topic, but is there anything else due this week other than the test? No, just check-ins and the exam are due for this week. Okay, so real quick before we go, 2 quick practice problems, and then we can stick around for questions if you have anything pressing for a little bit.
This does not count for check-ins.
No, you should email your TA or go to one of the help sessions that are left this week. Rachel has one tomorrow if you want to go.
Yeah, I do. 1 p.m.
And Sebastian and Jess will have one Friday morning also.
Yeah, they're having theirs in the morning before the exam. So if you realize Thursday night, like, holy shit, I don't know this, they're having them Friday morning.
Yes. Okay, real quick. What's the advantage of miRNA not being 100% complementary to mRNA in humans? Why wouldn't this be necessary in siRNA? It's probably a good turn on your mic question.
Isn't it because it prevents like wasting resources, since in some cases the mRNA can be used again if it's like sequestered to the P bodies?
Yep, it's definitely an advantage.
Um,
And yeah, go ahead.
oh, sorry. Another might be, since it's also used as a, what do you call it? A defense mechanism against like prokaryotes and like viruses, and since they mutate quickly, it could like give a longer shelf life to their like immunity.
Yep, that's definitely another good example. Yeah, so that's like kind of what I was going to say too. So if it's not 100% complementary, then it can target a wider variety of mRNA. So like you said, it'll let them be defensible against more different viruses. Okay, last question and then we're done. True or false, Drosha turns PRI mRNA into PRE mRNA in the cytoplasm. Yep, you guys caught the trick. It's nucleus, not the cytoplasm. All right, thank you guys for coming. We can stick around for a few minutes. It's getting late, so not too long if you have any questions. But here's some Huskies if you love Huskies like me.
And then here's the last side. Remember, you have 12 hours to do it. It should only take you 2 to 3 hours to complete. It's all open response, no multiple choice. Starts at noon Friday. Don't miss it.
Yep. Thank you guys for coming. We tried to make it as comprehensive as possible, so you guys have these while you're taking it.
You guys are gonna do so great. You're gonna do awesome.
You guys got it. You know more than you think you do.
Yeah, you definitely do.
Awesome review session.
Yeah,
Thank
and
you,
we will
guys.
send out the slides.
Yes. And I will upload it to YouTube now, so I don't get a billion emails.
Yeah, I want to end the recording.
Okay.
Oh, thank you guys. They said we did a good job.
Thank you. If you have questions about wording during the exam, you should email Laura.
Yeah, email Professor Francis.
She's the one that can answer exam questions. We really can't.
Oh, guys. They're so cute.
Took forever, so I appreciate it.
I can email the slides, Jason, if you want to do the recording part.
Okay. Well, wait for the recording to be. I can do it because I have to wait for the recording to be uploaded.
Oh, all right. You sure?
Yeah, because you need the link.
Okay. Yeah, that's fine.
All right. Thanks, Rachel. Does anybody have any
Yeah.
last minute questions? All right. I'm just gonna end
You're
it.
welcome.
Have a good one. That was fun.
I feel like. Wait, end the recording.
Did we?
No.
Where is the record? Oh.